Okay, we just finished G2. We got the green light. We're gonna go ahead. We're gonna divide. We're gonna make two identical daughter cells. That is the entire purpose of mitosis. We are making two identical cells. One cell will divide into two identical cells. These are the general phases of mitosis. So G1 as G2, that cell can spend a long time in those phases. These phases, mitosis, happens really quick. Some cells, 20 minutes maybe, right? We don't want to linger here in mitosis because the more we linger, the more chances we have that something's going to go wrong. We don't want anything to go wrong in mitosis. So we're going to walk through the general steps, okay? But the end goal here is we're going to take those sister chromatids that are attached to the centromere, and we're going to break them apart so that way we get two cells with an entire set of DNA. So let's get to it. These are the phases that occur in mitosis. This is the order in which they occur. I'm expecting you should be able to tell me the order and the big events of each of these phases. Okay? So mitosis is the division of the DNA in the nucleus into two. Cytokinesis is when we divide up the rest of the cytoplasm of the cell. Okay, so let's jump right in from the top. Prophase and Prometaphase. So our DNA has already been duplicated, right? This is where your DNA is in these X shapes. But now, and actually, let's go back and look at G2 phase. G2 phase, our DNA is all unwound because we're using it, right? We're using our DNA, so it's open. It's not super condensed and coiled up in G2. By the time we start mitosis starts with our DNA condensing. The only time we see DNA in nice little, tight little chromosomes like that are in the X shape is during mitosis. It's the only time we see DNA in this shape. Our organelles kind of scooch to the outside of the cell, to the edges of the cell, because they want to be out of the way. Mitosis is sometimes called the dance of the chromosomes. So they gotta get out of the way. They don't want to block those chromosomes from doing their thing. So organelles move to the side of the cell. Our centrosomes, remember those are the organelles that are going to control the cytoskeleton to move the DNA around. They get set up. Their location for this entire process, they're going to set up kind of poles on either side of the cell. You know how like on a football field you have a field goal on one end and a field goal on the other end? Kind of what the centrosomes do. They set up on either side of that cell, on opposite sides. Prometaphase is when we start to break down that nuclear envelope. We gotta break apart that DNA. We gotta move it around. We can't do that from within the nucleus. So we actually, during mitosis, DNA for a short period of time will be in the cytoplasm. It's still protected, but the nuclear envelope actually breaks apart and disappears during prometaphase. This breaking apart of the nuclear envelope allows the cytoskeleton components, the spindle fibers is what we call them, that are created from the centrosome. Those spindle fibers grow out of the centrosome and they attach to the DNA. So they grab onto the DNA, and that's what's going to allow the DNA to move around. So prophase and prometaphase, this is our kind of like setup phase. We are getting ready to divide that DNA in prophase and prometaphase. So DNA condenses, so that way it doesn't get all tangled up when we start to move it around. So it condenses into those nice little X-shaped chromosomes. Nuclear envelope falls away, spindle fibers start to grow out and attach to that DNA. Now, next, what's going to happen? This next phase, we call it metaphase. Meta means middle. So what's going to happen? So spindle fibers are going to push and pull and push and pull and push and pull on those chromosomes until they create one straight line. All 46 chromosomes in our genome in humans, one straight line down the very middle of the cell. And they're arranged like that. They're pushed and pulled by those spindle fibers coming out of the centrosome. So centrosome fibers connect the DNA, push and pull each chromosome until all DNA is in one long line in the exact middle of the cell. Our sister chromatids are still attached to each other. Remember, we call each half, so there's our centromere. Each of these, that's one sister chromatid, that's a sister chromatid. Remember, these are identical pieces of DNA. Sister chromatids are still attached. What do you think is going to happen? The next phase, anaphase, basically ANA means away. We are going to basically tug on either side of those sister chromatids and break them apart. So metaphase, we're lined up in the middle. Anaphase, these spindle fibers start to reel back in, kind of like reeling back in a fishing line. We reel back in those spindle fibers, and those opposing forces on either side of that chromosome basically separate those sister chromatids into two. Now, once those sister chromatids are separated, we call each a chromosome. Because remember, we count the number of chromosomes by counting the number of centromeres. So here's a centromere, here's a centromere, here's a centromere, here's a centromere, right? So now we actually have double the number of chromosomes in our cell than we did during metaphase. So sister chromatids separate during anaphase. They start to move to either end of the cell. They're being pulled by those spindle fibers that are coming out of the centrosome. And the cell gets a little bit oblong in order to make room for those chromosomes coming to either end of the cell. So anaphase separates sister chromatids and pull them towards that centrosome on either side of the cell. Telophase, telo means end. So here we got to wrap it all up. Literally, we're going to start to wrap it all up in that nuclear envelope again. So telophase is when we start to construct that nuclear envelope, that membrane that surrounds the DNA again. And we start to build two new nuclei. So this light kind of pale squiggly line is representing that nuclear envelope being constructed. The spindle fibers, they did their job. They moved the DNA, they separated the sister chromatids, they start to get broken down. But look at how the cell looks a little different here than it does here. See this little piece right here? This cell starts to kind of pinch inwards on either side. That process is cytokinesis. So we divided the DNA. The end of mitosis is telophase. Now we divided the DNA, created two new nuclei. Now we have to divide the rest of the cell. So now we have to divide the rest of the cytoplasm, all the organelles that are in here. Remember, they're kind of shoved off to the side. They're out of the way so that we didn't interfere with the DNA. Now, we got to go through cytokinesis. We got to separate those cytoplasms. So cytokinesis happens slightly different in animal and plant cells. Let's talk first about animal cells. So that kind of like furrow that was created, the little pinching in, call it the cleavage furrow. And basically what happens is that in the exact middle of the cell, there's a ring of proteins, ring of cytoskeleton proteins that are constructed that start to constrict, kind of like a belt. They start to cinch down, which basically causes that cell membrane to kind of fold inwards, and those proteins are going to constrict. That contractile ring is going to constrict until we finally create two new cells, basically. So contractile ring of proteins is contracted in the middle of the cell. Here's our nuclei, right? And that's going to contract and contract and contract until basically those membranes pop apart and we get two new cells. Remember, these are identical to the cell we started with. Completely identical, exact copies. Cytokinesis in plant cells, slightly different, but instead of constricting the membrane to create two new cells, we basically just build a new cell wall right in the middle of it. So we construct a new cell wall because remember, plants have a cell wall, animal cells do not. That cell wall is rigid. We can't bend it inward like we can in an animal cell, the membrane. So we just build a divider, basically, between those two cells, and that's how we get cytokinesis in plant cells. So now, ta-da! We have two new cells, two identical cells. We've done it. We've made it through mitosis. Now, what are these cells doing? What phase do they enter into? G1 phase, right? They start the whole thing over again. They start their journey over again, but they start with G1. So can you identify what phase the cell is in based on what's happening inside of it, based on what the DNA is doing, based on what the chromosomes look like? Right? This isn't a plant cell, okay? These are onion cells, but you should be able to tell me if I give you an image of a cell that is in one of the mitotic phases, you should be able to tell me what's going on here, right? Like this one right here. DNA is all lined up in the middle. What phase is that? Metaphase. What about this one? DNA kind of looks like little fingers getting pulled apart. Anaphase. What about this one right here? We can see the pieces of DNA, but they're still pretty much contained in the nucleus. That'd be a prophase. This one right here. DNA is not condensed. We still have the nucleolus. We can still see the nucleolus. Or like this one right here, DNA is not condensed, DNA is not condensed. If the DNA is not condensed, that's going to be an interphase. Okay, so the DNA is not condensed, interphase. See the difference between these ones and like this one right here? See the difference between those? Prophase, interphase. Okay. This one right here, DNA is separated. We're starting to build two new nuclei. This kind of yellowish stuff in the middle, that's a new cell that's being constructed, a new cell wall that's being constructed. So this would be telophase and cytokinesis. Okay, so you should be able to identify what phase a cell is in based on what's happening in that nucleus. So remember, this is all highly regulated. We don't want a cell to divide if something's wrong with its DNA, if it's not ready to, if it doesn't have all of its organelles in place. So it's a highly, highly regulated process. We have many fail-safes in place in our cells to prevent a cell from dividing when it's not ready to. If a cell proceeds when not everything was really prepared for it to go through this process of mitosis, can have some major issues arise. That's really how cancer comes about. Cells dividing without having any sort of check on their cell division. So we've talked about a few of these. We talked about there's a G1 checkpoint, making sure the cell is in a favorable condition to go through division. Is there enough energy? Is the cell damaged at all? Do we have enough cytoplasmic material? Do we have enough organelles? Are we ready to go? Synthesis phase and then G2 phase. The other major checkpoint is the G2 checkpoint. So is everything prepared for division? Is our DNA replicated properly? Are there proteins assembled? Or do we have two centrosomes, right? If we pass that checkpoint, we can then go into the mitotic phase. And there's actually another checkpoint during the mitotic phase, especially during metaphase. Here, the checkpoint is, did these spindle fibers attach correctly to those chromosomes? If they didn't attach correctly to the chromosomes, the DNA is not going to divide properly, and that's not good. We're going to get one cell with a little bit too much DNA and one cell with a little bit too little DNA if the spindle fibers don't attach properly. So this could trigger cell death, what we call apoptosis, scheduled cell death basically, if the spindle fibers are not attached properly, because we do not want a cell that has too few or too many chromosomes. It typically does not end up well. But you're like, well, but cancer exists. We have these checkpoints. There's at least 3 major checkpoints. How come we still get cancer? The answer is DNA mutations, basically. So cancer is uncontrolled mitosis, and there can be sometimes mutations in certain genes that help regulate those checkpoints. If there's a mutation in those genes, that checkpoint may no longer function properly, and so a cell could proceed past that checkpoint without actually meeting the requirements. So those genes that regulate those checkpoints, we call them proto-oncogenes. So these are genes that, if there's a mutation in them, that can cause cancer. So proto-onco are just genes that regulate the cell cycle and those checkpoints. After mutation, we call them an oncogene though. And so a gene that has gained a mutation and now is causing that cell to be cancerous and to divide rapidly without any check on its division. So we're going to look at the example CDK gene. The CDK gene, it's a proto-oncogene that if it mutates can become an oncogene. But what causes these mutations? Could be environmental factors, could be stress, could just be a natural mutation that arises. Every time our DNA replicates, there's a chance that there's going to be some mutations in there, right? But if this gene is mutated, cancer is highly likely. So the CDK gene codes for a protein called p53. And what p53 is doing is it's part of those checkpoints. It's a protein that is checking for DNA damage, checking for cell abnormalities. It's also sensitive to hypoxia, so low levels of oxygen. If any of those things are checked off, p53 detects DNA damage, if it detects cell cycle abnormalities, something's going on in the cell that's not right, if it detects hypoxia, it basically triggers a pause on that cell cycle. It says, nope, we gotta stop right here and fix whatever's going on. If that repair can't happen, then we trigger what's called apoptosis, which is programmed cell death. So cells have the ability to basically kill themselves off if they're at risk of becoming cancerous, or if they're so damaged that they're not functioning properly, they can trigger apoptosis. If there's a mutation to this gene that codes for p53, that protein, remember mutations in DNA end up in the mRNA, which mean that if there's a mutation in DNA and the mRNA, and that ends up in the amino acid sequence of that protein, change the amino acid sequence, that protein is not constructed properly, we change the structure of that protein, we change its function. So if there's a mutation in that gene that codes for p53, that p53 protein no longer functions properly because it's not constructed properly, because we don't have the correct sequence of amino acids in the correct order. Which means now p53 cannot check for DNA damage, it cannot check for abnormalities, it cannot check for hypoxia. It's broken, right? It's like that little gate in the parking garage. It's just busted. You can just go right on through. So the cell cycle continues. Even if there's DNA damage, even if the environmental conditions are not right, even if there's low oxygen, even if there's abnormalities in the cell, that cell cycle continues and the cell can become cancerous. Because p53 can't pause it, it can't stop it. There's many other genes and proteins that this isn't our sole guardian against cancer. There's many, many genes and proteins that function in this regulation of the cell cycle. This is just one of the better known ones. But the idea here is that mutations to proto-oncogenes can cause them to mutate into oncogenes, which means they are no longer able to regulate the cell cycle, and that cell can become cancerous. The good news is our immune system also can help detect cancerous cells. So cancerous cells will have abnormalities that display on the cell surface, which the immune system can detect, but sometimes our immune system can't detect them, or they replicate so quickly that they're kind of overthrown the immune system in a sense. Okay, so we have many layers of fail-safes here, but enough mutations are accumulated and that cell can become cancerous. So this is why often we treat cancer with medications or with specific treatments like radiation that triggers that are the most effective against cells that are currently going through division. So chemotherapy, often those chemotherapy drugs target some part of that mitotic phase. So, for instance, my dog had chemotherapy for a very treatable cancer, but the drug that she was given targeted the spindle fiber formation. So the spindle fibers can't form because this drug is messing with that process, can't divide. The cancer is basically stopped in a sense, right? Radiation is also most effective against cells that are actively dividing. So this is why one of the side effects of chemotherapy and radiation are hair loss, sick to your stomach, things like that, because those cells in our stomach and our hair are rapidly dividing. So we're getting the cancer cells, but we're also damaging healthy cells as well that just have a fast cell cycle. There's some new and exciting stuff going on, looking at how to program our own immune cells to attack just our cancerous cells. So we can train our immune cells to look for a very specific marker that is unique to that cancer, and then they would attack that specific cell type. So there's some new exciting stuff going on there as well. And there we go. I don't like leaving this on cancer, but the whole idea of mitosis is one cell divides into two exact copies. Two exact copies. Mitosis, one cell makes two exact copies, each with a complete set of DNA.